Hello, ladies and gentlemen. This is an egg, and you may not realize it, but an egg is just a single cell. So we are going to take a look at some of the organelles that we can see in this single cell. So this is the egg shell, and when you crack an egg, part of the membrane comes off within the shell. So if I peel this back, you can see some of the membrane, which all cells have. So this is part of our plasma membrane of this cell. If we look at our egg that has been cracked, you can also see that we have egg white, okay? And egg white is cytoplasm. All cells are going to have cytoplasm. So if you've ever cracked an egg, then you know what cytoplasm feels like. It's this goopy material. And this is where a lot of chemical reactions are going to happen in the cell. You also have the yolk. And the yolk is the nucleus of the cell. So this is where the DNA is going to be kept. And in an egg, you only have half the DNA that you would need to make a whole organism. So yolk is your nucleus. Now, if you've ever heard that people with high cholesterol should not eat this nucleus, this yolk, there's a reason for that. The yolk is made of lipids. And if we think back, lipids are fats. So this is where a lot of the energy is going to be stored for the growing organism, in this case, a chicken. The egg white, the cytoplasm, is made mostly of protein. So that's why the cytoplasm is healthy for you, and they encourage you to eat lots of cytoplasm or egg white to build good muscle. If we draw a cross section of an egg, this is basically saying that you take an egg and you cut it in half and it all stays together, and then you can look at what pieces that you would have. So if we think about the outside, this is our shell, our outer covering. Inside of that, you're going to have that plasma membrane. And you saw me pull that out when I pulled that piece out of the eggshell. The goopy stuff in the middle is going to be your egg white, which is called your cytoplasm. And again, this is made of a lot of protein. That's why it's very healthy for you if you want to build good muscle. And the inside is your yolk, which is our nucleus. And the yolk is made of a lot of lipids to provide energy for that growing offspring, and is also where the DNA is going to be held. So this is our cross section of the egg. The egg shell is made of a substance called calcium bicarbonate, and this happens to be the same substance that seashells are made of, and that's going to come back into play here in just a minute. So I need to get this eggshell off if I'm going to work with the cell itself. So I'm going to use vinegar, and vinegar is an acid, which means it has a pH of less than 7. So here's some vinegar, and let's see what happens when I pour it onto my egg. You can see that we have lots of bubbles forming around the shell of this egg and coming up through this vinegar as the vinegar is eating away at this eggshell. Now, carbon dioxide is a chemical in our atmosphere that's becoming more and more prevalent. As we cut down trees for deforestation, we're taking away the trees that are doing photosynthesis and pulling this CO2 out. At the same time, we're still burning a lot of fossil fuels, which is pumping that CO2 into the atmosphere. The more CO2 that enters the atmosphere, the more there is to come in contact with ocean water. And when CO2 comes in contact with ocean water, it becomes carbonic acid. Now remember what I told you about eggshells and seashells. Both of them are made of calcium bicarbonate. And we can see that that acid is going to eat away at this calcium bicarbonate. So what do you think might happen to our seashells as our oceans become more acidic? Hopefully you're thinking, well, those sea creatures that secrete those seashells are not going to have as much protection because that seashell is going to slowly be eaten away. And if you've ever gone to the beach and you find seashells that have little holes drilled in them, that's usually caused by a type of snail, a mollusk, that climbs over top of these sea creatures and secretes an enzyme that drills this little hole into the seashell, and then they can suck out that organism. So it's going to make them much easier prey if they don't have that shell there to protect them. So I have some eggs that I've been soaking for several days in vinegar, and now if I just wipe this egg, you can see that the shell is just coming off like a powder. So I'm going to go rinse these eggs off and get this shell off, and we'll be down to this membrane that I showed you earlier and it will be intact, so it's still going to hold this egg all together. All right, I have washed off my eggs, and now this is what they look like. This is the first egg. See, they're rubbery, really mushy feeling. This is the second egg. We've got them all down to their membranes now. And remember, membranes control what comes in and out of the cell. And then this is the third egg. So in this lab, we're going to be looking for how these membranes control what passes back and forth between these eggs. So we're going to be putting them into different solutions. You see those on the board: water, syrup, and air. Okay, so I've got a cup for each different solution. I've got one egg in each cup. Now, before I put these eggs into their solutions, I'm going to want to weigh them, because I want to know what their mass was before I put them into the solution, so I can see how the solution affected their mass afterwards, and then hopefully determine what goes across this membrane. So I'm going to weigh my eggs, and here I have my scale, and I will record those weights behind me on the board. Now, I do want to make sure that I'm just weighing the egg. I don't want to be weighing the water or the vinegar. So I'm going to dry it off. And my water egg weighs 69 grams. So I'm going to go ahead and record that on the whiteboard. All right. Now I'm drying the egg that I've designated to go in syrup. Again, want to make sure that I weigh the egg, not the water on the egg. And it weighs 78.8 grams. It seems to be going back and forth between those two. So, well, it's settled on 78.7. So the syrup egg, 78.7 grams. And the last egg we have is for air. And so we're going to dry it off and weigh our egg that's going in the air. And it's 66.9 grams. Okay, I have my two solutions, water and syrup, and you can see that I have 100 milliliters of each of these. So in the water egg, I'm going to pour my 100 milliliters of water. In my syrup egg, I'm going to pour my 100 milliliters of syrup. Now I'm going to use a spoon to scrape this syrup into the egg. Now keep in mind, I want to use the same amount of both solutions because I want to control what's going into my egg experiment. See what I did there? The air egg, I'm going to leave alone because I want to see how air affects the mass of my egg. I'm going to cover these eggs and I'm going to leave them alone for the next day. And tomorrow I'm going to come in, I'm going to wash them off, I'm going to reweigh them, and we will see how these solutions have affected the mass of these eggs and try to determine what has moved across the membrane. We are back for day 2 of our experiment. So just a reminder, yesterday our water egg weighed 69 grams, our syrup egg weighed 78.7 grams, and our air egg weighed 66.9 grams. Today we are going to look at these eggs' appearance and their masses to see if those masses and appearances have changed. So I've already washed and dried these eggs, and we'll start with your water egg. This is the water egg. It's really full. It feels like it is about to pop. This is our syrup egg. It is deflated like a balloon that has lost air. And this is our air egg. It still feels pretty full as well. So now we're going to turn our screen to your scale, and we're going to see how much these eggs weigh. All right, and I've already dried this, but we'll dab it just a little more. Be sure that we are not weighing any water. And before I start weighing these eggs, if you have not made your hypothesis about what you think has happened to these eggs in the last 24 hours after soaking in these solutions, please make sure that you do so. You can pause it if you need more time. So our water egg today weighs 69.2 grams, which is a little more than it weighed yesterday. Our syrup egg weighs 40.9 grams, significantly less than it weighed yesterday. And our air egg weighs 65.9 grams. And it has lost a gram since yesterday. All right, so what we have seen, our water egg that was sitting in water got bigger. Our syrup egg that was soaking in syrup has gotten smaller. And our air egg has only lost 1 gram, okay? So let's take a look at our solutions as well. This is the syrup from yesterday. If you can see, it's very watery now. It's got a layer of liquid on top of that syrup. And as for your water, it's still in there, but it is less water than was in our cup yesterday. So, what do you think may have passed through our membrane? Well, judging by the fact there's less water in the water cup, there is more solution in the syrup cup, and it is more watery in appearance. And there is actually a tiny, tiny bit of water in our air cup. We can assume that water has moved across this membrane. The syrup did not. So let's think about why that would make sense. A phrase that I want you to remember is that salt and sugar suck. And what we mean by that is wherever there is more salt or sugar, it's going to pull water in that direction across a membrane. Okay? So if we think about the water egg, there was more water in the cup than there was in this egg, and so water moved across the membrane. Now, we didn't see a lot of water move across the membrane because this egg had already been soaking in vinegar, which has water in it as well. So we can assume that some water has already passed through this membrane, and the concentration of water wasn't that different between the egg and the watery environment it was placed in. If we think about the syrup egg, though, there was a lot of water in this egg yesterday, and significantly more water in the egg than there was in the syrup. So the water flowed down its concentration gradient, or it went toward where there was more salt or sugar. So wherever there's more salt or sugar, there's going to be less water. Now our air egg, we may notice, didn't really change a whole lot. It's not in a solution, so it's not going to have water necessarily pulled one way or the other, but we can think about why the shell on the egg might be very important for our egg to survive on a dry land environment. We'll talk about that a little bit more tomorrow when we weigh these eggs again. So I'm going to rinse out this water and syrup cup, and we are going to set this lab up a second time, but we're going to switch the eggs. So the egg that was in syrup yesterday is going to go in water. The egg that was in water yesterday is going to go in syrup. And we're going to see if this process is reversible or if this membrane is now done. Okay? So I want you to make your hypothesis for part B. Do you think that these eggs will switch appearances? Do you think the water egg will now deflate and the syrup egg will inflate? Or do you think this membrane is ruined and it can't do this in two directions? So I want you to think about that. I'm going to clean these cups out and we're going to set this experiment up again. I now have clean cups, so I'm going to use my water cup and I'm going to put my syrup egg in the water cup. So it's now become my water egg. I'm going to take my water egg and I'm going to put it in this syrup cup because it's now my syrup egg. And my air egg I'm going to leave alone. So in the water cup, I'm going to pour 100 milliliters of water, same as I had yesterday. And in my syrup cup, I'm going to pour 100 milliliters of syrup, same as I had yesterday. And we want to make sure that we use the same amount of water and syrup because we want to have as much control over this experiment as possible. So now I'm going to cover my cups just like I did yesterday, and we'll check them again in 24 hours, and we will find out if these membranes are destroyed, if their properties have been destroyed, or if we can in fact change the masses of these eggs and reverse their appearances. We're now on day 3 of our experiment, so just a reminder, this was our chart with all the masses of our eggs. This was the masses of our eggs on day 1, and these were our masses yesterday. So we can see that our egg soaking in water went up just a little bit, and we can attribute that small gain to the fact that the concentration of water in the egg and in the actual water itself were not that different. That egg had been soaking in vinegar for a while, so there's a good chance it had taken up some water from the vinegar and had already gained some water mass. We have our syrup egg that had lost a significant amount of mass, and then we had our air egg, which had only lost 1 gram. So yesterday we switched our egg that was soaking in water and we put it in syrup, and we took our egg that had been soaking in syrup and we put it in water. So we're going to have to change this chart around a little bit. These are now going to be the masses that we had yesterday. So our air egg is at 65.9 grams. Since we switched our water and our egg soaking in syrup, I'm going to take the mass of my former water egg, which was 69.2, and this was the mass that the egg soaking in syrup today had yesterday. I'm going to take the mass of my syrup egg from yesterday, and I'm going to put it in the water spot, because this was the weight of my egg yesterday when I put that egg in water. Okay, so we've switched them. Now I'm going to go wash off my syrup egg. I'm going to dry my eggs, and we're going to reweigh them. Please make sure you've written down your hypothesis for today. We were thinking about whether or not this membrane is still able to allow materials to pass through it. So do you think that former water egg that we have now soaked in syrup, do you think it could have lost mass? And do you think that the former syrup egg that we placed in water could swell back up and gain mass? So if you've not already done so, please make sure that you've written your hypothesis for this part of the lab. And here are our eggs for today. This was our former syrup egg that we soaked in water. This was our former water egg that we soaked in syrup. You can see it's very deflated. And this is our air egg. And you can just kind of compare the sizes of these 2 eggs. This is air and this one's been in water. And now we will get our masses for these. Remember that we dry off our eggs to make sure we're not getting any extra water weight. So our air egg is down to 65.8 grams. It's not lost a lot. Our new syrup egg is down to 32 grams, even. And our new water egg is 78.8. So here are our masses from today. Our egg yesterday weighed 40.9 grams when we put it in water, and today it weighs 78.8, and we can see it is obviously swollen back up. We have our syrup eggs mass yesterday coming in at 69.2 grams, and today it's gone down to 32 grams. And we have our air egg at 65.9 yesterday, and today it is at 65.8. You can also see in our syrup cup that the syrup is very watery. It looks like it has a layer of water on top of it. And the water in our water cup has gone down significantly. It's down to about here, and was up to about here. So we can see here that our plasma membrane is still working. It is allowing water to cross it, and we have evidence for this in our masses. When we put an egg without a lot of water in it into water, water flows down its concentration gradient, so that water is going to go from outside of the egg to the inside of the egg.
When we put an egg with a lot of water in a solution that does not have much water, like syrup, that water is going to flow down its concentration gradient and it's going to go out of the egg from where there was a lot to where there was a little. It's going to cross the membrane. You can also remember this by salt and sugar suck. Wherever there's more salt and sugar, there's less water. So the water is going to go toward the salt and the sugar. With our air egg, you can see that it lost just a little bit of mass, and we'll come back to that here in just a few minutes. I do want to talk to you about a couple of other things. So think about what would happen to your blood cells if you drank a lot of water, like what happened with Jennifer Strange. I had you read that news story. So when you drink a ton of water and your blood has more water in it than your blood cells have in them, then you can expect that water to flow from your blood into your blood cells, and they can in fact swell so much that they pop. Now with the case of Jennifer Strange, that water in the blood is also going to the brain, which is going to swell the brain cells, and that's going to put pressure on the brain stem and cause death, which is what happened in her case. Now, what do you think would happen to your blood cells if you drink a lot of sugary drinks? If your blood starts taking on a lot of sugar and there's more sugar in your blood than there is in your blood cells, then you can imagine that your blood cells are going to shrink because they're being placed in a sugary environment. And so they're going to shrivel up, maybe not be able to carry oxygen quite as well. When it comes to the air egg, I want us to think about organisms that lay their eggs in water compared to those that lay their eggs on land. Well, we can see that if an egg without a shell is laid on land, it's going to lose mass. This egg actually started at 66.9 grams and is now down to 65.8 in just a couple of days. So without a shell on it, that egg is going to dehydrate and probably will not have enough water to allow the organism developing inside to reach maturity to where it could actually hatch. So that's why we have amniotic eggs for organisms that live on land. This shell is there to help hydrate them. It's not so much for protection. It does offer some protection, but eggs are still pretty fragile. I mean, you can break an egg pretty easily and get to what's inside. So the shell is there in a lot of ways to help these eggs stay hydrated. Now, think about organisms that lay their eggs in water, like fish and frogs. Those eggs do not have a shell on them. They are laid in the water, and so that water can freely flow into that egg and keep it hydrated. Now, I hope we learned something from your egg experiment. I think it is an excellent way to make all of this make sense. And we're going to talk about this and tonicity a little bit more in your lecture. So we'll get some names for these types of solutions that we have placed our eggs in. See you later.